Dr. V.K. Barun from Medico Point, Patna, and today class is the digestion of food. So what is digestion of food? That is the breakdown of food is called digestion. So the digestion mainly takes place in the mouth, a stomach, and a small intestine. No digestion takes place in the liver because liver has no any enzyme. Because liver has only bile, it helps in the digestion. And the pancreas, there is no any digestion in pancreas, but pancreatic juice releases and the help in the digestion in a small intestine. And there is no any digestion in large intestine. So in large intestine only mucus is present, so there is no digestion. In esophagus, no digestion. So in esophagus, liver and large intestine, there is no digestion. The where digestion takes place? So digestion mainly takes place in the mouth, a stomach and a small intestine. So first of all, mouth. So in mouth, digestion of the carbohydrate takes place. That means a starch. So a starch converted into maltose with the help of salivary amylase. Because the mouth consists the salivary gland, that is parotid gland, submandibular gland and sublingual gland. So these three pair of gland present in the mouth, buccal cavity. So it releases the salivary amylase and lysozyme, which is present in the saliva. So lysozyme is antibacterial enzyme which kills the bacteria, and salivary amylase, that is pH is around 6.8, so it converts the starch into disaccharides such as maltose, isomaltose and limidextrin. So this is the digestion of food in mouth. Second is the digestion of food in stomach. So stomach consists the gastric juice, and it consists gastric gland, and gastric gland produces gastric juice, and it has three type of cell. First is mucous neck cell, this releases mucus. Second is peptic cell or chief cell. The peptic cell or chief cell or zymogen cell, it releases pepsinogen या propepsin, pepsinogen or propepsin, that is in inactive form. And second is parietal cell or agentic cell, which releases two things. First is HCl and second is Castle intrinsic factor. These factors, intrinsic factor helps in absorption of vitamin B12 and HCl. So this pepsinogen is in active form, and this pepsinogen is in active form and converted into pepsin with the help of HCl. And this pepsin acts on the protein, and this protein converted into proteases and peptones. So this is the function of HCl, and HCl also provide the media acidic. So the pH of gastric juice is around 1.8 due to HCl. And second is renin. First is the enzyme is pepsinogen, second is renin, and third is lipase, but very less amount of lipase. So the renin convert the milk protein, mainly casein, converted into para-casein, and this para-casein with the help of calcium to convert into calcium para-caseinate, and it helps in curdling of milk. And this renin is only present in human being, in infant and in ruminant, that is in calf. And very less amount of gastric lipase is also present, which help in digestion of fat and converted into fatty acid and glycerol. So this is the overall digestion in the stomach. And third part is a small intestine. So digestion is a small intestine, and the intestinal juice is succus entericus, and the pH is around, intestinal juice pH is around 7.8 due to pancreatic juice and intestinal juice. So the pancreatic juice also consists enzyme, that is 6 type of enzyme present in the pancreatic juice, such as trypsinogen, procarboxypeptidase and chymotrypsinogen. These three are inactive form. And fourth is amylase, pancreatic amylase, then lipase, that is also called as trypsin, and pancreatic amylase is also called amylopsin, and nuclease, that is DNA age and RNA age. So total 6 type of enzyme present in pancreatic juice. So pancreas not directly involved in the digestion, but pancreatic juice helps in the digestion in a small intestine. And large intestine there is no digestion because there is no any enzyme, only mucus is present, and it is protective and helps in the lubrication of food. These a small intestine also consists around 9 type of enzyme. So first is enterokinase, second is ellipsin, that is aminopeptidase, and dipeptidase, lipase, maltase, sucrase, lactase, nucleotidase and nucleosidase. These are nucleotidase, nucleosidase. So nucleotidase and nucleosidase present in the intestinal juice, that is succus entericus, but nuclease present in pancreas. So this is very different. Nuclease present in pancreas, but nucleotidase and nucleosidase present in succus entericus, and nuclease present in pancreatic juice. So 6 type of enzyme present in pancreatic juice, and total 9 type of enzyme present in succus entericus, intestinal juice. And pH of intestinal juice is 7.8 due to bile. And bile consists the sodium glycolate, sodium tarocholate, that is bile salt, and sodium carbonate. And bile pigment is bilirubin and biliverdin. So the bile pigment and bile salt present in the bile, but bile has no any enzyme. So the first is enterokinase of the succus entericus. So the succus entericus consists enterokinase, which acts on the enzyme, not acts on the directly on the food. It acts on the enzyme. So it converts the inactive enzyme to active enzyme. So enterokinase acts on the trypsinogen and it converts into trypsin. So inactive trypsinogen convert into active trypsin. And this active trypsin acts on the chymotrypsinogen and procarboxypeptidase. And both chymotrypsinogen and procarboxypeptidase in inactive form convert into active form. And these three means trypsin, carboxypeptidase and chymotrypsin directly acts on the protein. And proteases and peptone, and it converted into dipeptides. So it is very important. And next is amylase, that is amylopsin. It acts on the starch and convert into disaccharide. And lipase, that is the astepsin, it acts on the fat and converted into fatty acid and glycerol. And nuclease, that is DNase and RNase, nuclease present in pancreatic juice, it acts on the nucleic acid and convert into nucleotides. So this is very important. And the next is the intestinal juice, that is succus entericus. First consists the enterokinase. And next is ellipsin. Ellipsin, that is aminopeptidase. Actually, aminopeptidase and carboxypeptidase is collectively called ellipsin. So this is 3 terms. One is ellipsin, another is strepsin, and another is amylopsin. So pancreatic amylase is amylopsin, pancreatic lipase is strepsin, and aminopeptidase and carboxypeptidase is collectively called ellipsin. So strepsin, amylopsin, ellipsin. So ellipsin present in succus entericus. It acts on the dipeptides and convert into amino acid. And next is these dipeptides, which is formed by the proteases, peptones and protein. And these dipeptidase converts dipeptide into amino acid. And the lipase acts on the lipid and convert into fatty acid and glycerol. Maltase, this is the disaccharide. Maltase acts on the maltose, convert into glucose and glucose, 2-molecule glucose. And the sucrase acts on the sucrose to convert glucose and fructose. Lactase acts on the lactose, convert into glucose and galactose. So these are the 3 disaccharide: maltose, sucrose, lactose. And last is nucleotidase and nucleosidase. So the nucleotidase acts on nucleotides and converted into nucleosides, and nucleosidase acts on nucleosides and convert into nitrogenous base and sugar. So these overall are the product. So final product of fat is fatty acid and glycerol, carbohydrate, that is simple sugar, that is glucose and fructose, and protein, that is finite unit is amino acid, and then after digestion this goes into absorption. So this is overall digestion of food. So digestion only takes place in buccal cavity, stomach and small intestine. No digestion takes place in liver, pancreas, esophagus and liver. So this is overall digestion. Thank you.